Membrane and cell wall selective fluorescent dyes are important tools for the analysis of organelle dynamics in living fungal cells. Our protocols provide essential theoretical background and practical guidelines for the application of a selection of these dyes as truly vital stains. The key point for this is to use very low dye concentrations that do not cause cellular artifacts related to saturation or dye toxicity, while still providing good signal-to-noise ratios for high-quality long-term live cell imaging. With colleagues from the Department of Nuclear Medicine of the Medical University, we recently used these staining techniques to develop a novel approach for image-guided diagnosis of aspergillosis. The ability to monitor membrane and cell wall structures in real time was crucial for determining the uptake dynamics of the experimental drug compound.
To begin this procedure, first obtain a prepared preculture. Use a sterile scalpel to cut a small agar block that carries non-sporulating mycelium from the colony edge of the preculture. Place the agar block at the center of a fresh solid medium plate to inoculate the experimental culture. Incubate the experimental culture according to the developmental stage intended to be incubated.
Sample preparation is somewhat delicate, and optimization of image acquisition settings is rather complex. Visual demonstration of both procedures, accompanied by verbal explanation, greatly facilitates replicability.
To mount the